Should we start, Deepak sir?
Yes, I can
Hello?
hear you.
Yeah.
Hello? Sir, is my voice audible?
Yes, yes. You can
Yeah,
start.
yeah.
Good morning, one and all. I, Miss Ankita Shinde, Assistant Professor, Department of Pharmaceutics, DPU Pharmacy, Pimpri, Pune, feel privileged to welcome you all on behalf of DPU Pharmacy for the third session of Third International Faculty Development Program on Emerging Trends in Phytopharmaceutical Research. In the last week, we had our second scientific session on 18th March. It was delivered by Dr. Manoj Jadhav, founder and CEO, Isha Therapeutics, New Jersey, USA. He addressed the participants about the US FDA for several pre-IND meetings at the Division of the Anti-Infectives, Cardiovascular, Renal, CNS, Dermatology, Oncology, etc. Today, we have with us Dr. Deepak Kasote, Senior Research Specialist, Missouri University Metabolomics Center, University of Missouri, Columbia. He would be delivering this session on applications of metabolomics in herbal drug standardization. Now, I request Dr. Maruti Shelar to please introduce our today's speaker. Please, sir.
Maruti sir, please unmute.
Very good morning to one and all. I welcome Dr. Deepak Kasote sir on behalf of management, principal, staff, students, and all the participants into the third international FDP, Emerging Trends in Pharmaceutical Research. Dr. Deepak Kasote sir, currently working as a senior research specialist, Missouri University Metabolomics Center, USA. Dr. Deepak sir has completed his graduation and post-graduation from Shivaji University, Kolhapur, and PhD from Savitribai Phule Pune University, Pune. Sir is having more than 10 years of postdoctoral experience in multidisciplinary research areas such as plant science, natural product chemistry, phytochemistry, food chemistry, bioanalytical chemistry, nutritional biochemistry, nanoscience, and pharmacology. His core research expertise and interest in mass spectrometry and metabolomics. Sir has held various positions and honors at national as well as international level. Few of them: research scholar at North Carolina State University, USA, postdoctoral fellow, International Rice Research Institute, Varanasi, India, postdoctoral research associate, Texas University, USA, research fellow, Robert Institute, UK, research associate in South Korea as well as in South Africa. Sir has received many awards and recognition. Some of them: Young Scientist Startup Research Award from SERB, India, Postdoctoral Research Associate Fellowship, Republic of Korea, Postdoctoral Fellowship, Hungarian Scholarship Board, Hungary, Postdoctoral Fellowship, University of Technology, South Africa. On his credit, there are more than 40 publications in the journals of repute, more than 5 book chapters, and having Google citations more than 2,111. With this brief introduction, I request Dr. Deepak sir to enlighten us on the topic application of metabolomics in herbal drug standardization, with the aim to explore and understand the current technologies, methodologies, and practices in phytopharmaceutical research. Please, sir.
Thanks, Dr. Shelar, for introducing me. We will straight away start my presentation. Can you see my presentation?
Yes, sir.
Okay. Good morning to all. First of all, before going to start my presentation, I would like to thank the organization committee, principal, vice principal, Dr. Bhujwal sir, for inviting me to give a talk on this at this third international. Just a minute. Sorry.
Audible, sir, you're audible.
Yeah. So in today's presentation, my topic would be the application of metabolomics in herbal drug standardization. In this topic, I would like to give a brief introduction about metabolomics, herbal drug, what are the issues with herbal drug standardization, and then we'll see some research examples where metabolomics has been used for standardization of herbal drug. So already Dr. Shaila has provided my introduction, so I'll not talk about that straight away. I'll start with my presentation. So first phase of this presentation, I would like to talk about the current global scenario of herbal medicine and where we are as India and what are the challenges in its quality control. Then in a next phase, we will see details about metabolomics, what are the practical things involved in workflow of metabolomics, including some technical details. And as I said earlier, we'll see some we'll see some scientific research related to metabolomics, we generally use in herbal drug standardization. Just a minute, I have some issue, I think. I don't know what's happening.
Sir, please unmute.
Can you hear me now?
Yes.
You may agree with me that market for herbal drug, including including herb-based health supplements, are growing throughout the world. As a result, you can see the market for herbal drug is growing at the rate almost 20% a year. And it has been estimated that this market will reach up to the 350 billion by 2019. The figure shown by World Bank is really encouraging. And according to them, this world trade related to the medicinal plants and related product will reach up to the 5 trillion by 2050, which is the present day economy of Japan, which is the third largest economy in the world. So this is the market-based scenario of herbal drug throughout the world. But when we say this is global scenario, where India is. Sorry, I cannot go ahead. Just a minute. Hello.
Yes, sir.
I think there is some issue. I cannot go ahead. So what I will do,
Can you unshare it and again share the PPT?
Yeah, I think so. Can you see my presentation now?
Yes.
I don't know why it's not going ahead. Okay. Can you see my screen now?
Yes, sir. Yes.
Okay. So I don't know what's happening.
Sir, can you mail your presentation here so that we will do it from here?
So
Yeah, I will do. I'll email you.
huh. Sir, can you just try by without full screen of your presentation?
Oh, okay, I will do that then. I don't know why it's happening, but something is wrong. Now it's going. Let's see.
And now it is moving ahead.
Yeah. So now we just saw the global scenario of herbal drug market. Then now we'll see what is the status of India in the global scenario. So we are saying we have long use and practice of herbal medicines, but the figures which we saw about the Indian stand in global market are really disappointing. This is the news article published in Economic Times. According to them, they got this information from government resources. They said that the Indian share in the global market is just 0.5%. So you can understand that though global market is huge, but Indian share in the herbal medicine or herbal medicine market is really low. So this mainly because in India, we are not focused or we are not invested a lot in the research and development activities about the herbal drugs. Whatever knowledge we got from Ayurveda, Unani, Siddha, we are straight away using, and based on that, we are selling our product. But that the main reason our herbal drug industry is facing big problems, and mainly issues related to like quality, safety, and efficacy. So compare to us, if you look the Chinese traditional medicine, this is also one of the oldest and long use history medicine. It's also equally popular like our Indian medical system like Ayurveda, and their share in the global market is almost 20 to 50% compared to ours. And this mainly because China has done a lot of research on this aspect and mainly related to safety, efficacy, and quality of their drug. And that is the main reason they have this big market share in the herbal medicine industry. And it's not that just a figure. I have seen this figure things in abroad as well, in US, UK. If you go to any pharmacy shop, you will find Chinese medicines easily, and there are a lot of Chinese medicine on shelf. But compared to this, Indian products are really small. One or two products only I have seen, such as herbal medicine is the only Vidania somnifera is the very common product you will see in US from Indian origin. But about after that, there is nothing. So to understand more about this, especially comparative understanding about Chinese and Indian medicine, I don't know why again facing this problem. It's not going ahead.
Sir, just you remove the full screen
Okay,
and
then we'll
then
see
I
it.
think slides can be moved.
So earlier speaker had a same issue, or
No, no, sir.
I don't know why then happening this. Let me see. And
Or you can just email me, I will share from my end.
yeah, that also I can do. Just a minute.
So email to ravindra.wawale.
Yeah. Sorry for inconvenience, but
No issue.
we cannot do anything. Okay, I'll send it to you. Just check. Did you receive?
I-16.
Yes, I shared it. No problem with them. Presentation.
Okay. So can you move to next slide? Next.
No, I think there is a issue in slides, so I will share it again. Sir, is it okay in this format?
Yeah, it's for me it's okay, but I'm not sure about others.
I will make it bigger.
Yeah,
Yes.
Yeah, this is okay.
yeah, you can close that one.
On which slide, sir? Slide number?
Go to slide number 7.
Okay. Is this right, sir?
No. One above.
Yeah, that's fine. Okay.
Okay, sir.
Go to next one.
This one?
Yeah.
Okay, sir.
So I think it's not full screen.
No, slides are not moving on the full screen, sir. So we
Okay.
made it. Yeah.
So just now we saw that the Chinese share in the global market is almost 20 to 50%. So as I said, it's mainly because they have done a lot of research and development activities related to Chinese product. And now we'll see the comparative of Chinese medicine and our traditional medicine system in India. So if you see the Chinese traditional system, there are almost 1 million formulations. In terms, it's almost 10 lakh formulations they have, but they have formulations from different origins, such as from plants, animals, and there are some ethnic drugs, folk medicines. However, in that 11,000 formulations, there are some for almost 10 lakh formulations, almost 11,000 formulations are from medicinal plants. And out of that, they are exporting almost 8,000 varieties of Chinese origin or the herbal products to the almost 130 countries. Can you go to next slide? Compared to this, if you see in India, we have almost reported plant species as 45,000. Out of that, nearly 3,500 plant species are reported to have medicinal values. And out of this, 3,500 medicinal plants, nearly 500 to 700 medicinal plants are routinely used in Ayurvedic formulations. And if you see the total export of herbal, like Indian herbal export, you will see that only we are exporting 3% of as a herbal drug, and most of our export include 70% raw material and some plant extract we are exporting, which are not as a drug we are exporting as a health supplement such as turmeric and so on. Can you go to next slide? So this all facts indicates that we as an Indian need to spend a lot of time and money to investigate our herbal drugs is in the context of their safety, efficacy, and quality. This is the need of time. Can you go to the next slide? So what is exactly mean by the quality of herbal medicine means? What exactly we need to understand when we say quality of herbal medicine? There are several issues where we need to understand the quality of herbal medicine, especially what is the exact species, what exact part we need to use as a drug, and so on. So especially in our area, especially in India, we don't have that sort of information, or we have not generated that much data. So I'll give an example of Tinospora cardiophilia, which is we call as a it's a gulwel, but I don't know what call it in Indian other word, but in Marathi it is called gulwel. The Tinospora is a shrub. It's growing on different plants for support, such as Zatirrhita indica, acacia. So, and in our study, we also found that the Tinospora growing on different plants have different biological activity, especially immune modulatory activity. So we need to understand this sort of issues as well. Means there are several herbs. We are using several medicinal plants as a herb, shrub. So their quality according to growing locations, that also we need to define. And there is a big challenge in the Indian herbal industry is the adulteration and substitution. So that issue also we need to address in the context of quality of herbal medicine. I'll give an example of Tinospora cardifolia again. There is one species, Tinospora cyanosis, which is always substituted and adulterated with Tinospora cardifolia. And there are researchers, they are saying that the Ayurvedic description of Tinospora cardifolia, which matches with Tinospora sinensis, but still people are saying Tinospora cardifolia is the real species. And in our study, we found that, interestingly, Tinospora sinensis has more bioactivity, like hepatoprotective activity, compared to a well-known drug Tinospora cardifolia. So this sort of issues also we need to go in a deep and need to address. And for that reason, we need to do a lot of research related to the standardization of herbal drugs. So what standardization exactly means? Can you go to next slide? In standardization is a process in which set of parameters are used to define identity and quality of drug using various parameters like morphological, microscopic, physical, chemical, and biological evaluations. In the standardization of herbal drugs, several steps are involved, such as taxonomic identification and authentication, organoleptic examination of raw material or raw drug, micro and molecular examination, and in addition to that, some chemical composition analysis is also carried out by using TLC, HPLC, and DNA fingerprinting. And some of the claims are generally validated by using biological activity. But if you see these presently known quality control methods or herbal drug standardization methods are not providing any information about location of a drug, or whether that means even they don't provide any other biological or chemical markers for its quality control. Can you go to the next? So there are several limitations for the traditional methods. So nowadays it's generally recommended throughout the world or even in India to use advanced methods like metabolomics and chemometrics for the standardization of herbal drugs. These methods are really helpful for taxonomic identification and authentication because just a minute. Because in traditional methods, we require really specialist people to add taxonomic identification. But with metabolomics, any analytical chemist can do that. In addition to that, the present advanced methods, they are providing more information on what factors generally influencing the quality of herbal drug. And this modern approaches also helps to identify novel markers, especially when issues arises about the herbal drug substitutions and adulteration. This technique can be also helpful for geographical identification of a drug, because some cases we need to know exact from where a particular drug is coming, because efficacy is linked with geography as well. And as I said, this technique has a lot of significance in adulteration and substitution identification. So altogether, this advanced chemometrics, metabolomics and chemometric techniques has huge application in herbal drug standardization. Can you move to the next slide? So after understanding the state of art of herbal drug, its quality and significance of metabolomics, we'll now move towards the metabolomics. So metabolomics is a broad term generally used to define the set of metabolites. As you know that metabolite is the end product of cellular process, and that metabolite are not fixed, that are constantly changing. So analysis of all sets of metabolites is called metabolomics, and according to what sort of metabolites you are identifying, they are further classified into glycomics. If suppose you are identifying glycosides, then it's called glycomics. If you are identifying lipids, then it's called lipidomics, and so on. Generally, why we analyze metabolomics? Because it gives entire snapshot of physiology, or it gives the entire information about its chemotype in case of herbal drugs. Can you go to the next slide? Generally, there are two types of metabolites we analyze in metabolomics. They're called as primary metabolites and secondary metabolites. Primary metabolites are generally linked with energy metabolism of our body or plant. They require for normal growth, development, and reproduction, which mainly includes sugar, nucleotide, amino acids, proteins, and lipids. And the secondary metabolites are the another class of metabolites which are not required for survival of plant, but they are involved in plant defense. Like plant, they don't produce antibodies, they produce secondary metabolites for their own defense. So they have their own bioactivity, and we are harnessing that bioactivity as a drug for our welfare. These secondary metabolites mainly include ketone, aldehydes, steroids, terpenes, alkaloids, phenols, and so on. So in metabolomics, we identified both primary and secondary metabolites according to our interest. Go to the next slide. In metabolomics workflow, there are four main steps are common. First one is sample collection. After that, sample preparation, data acquisition, and data analysis. We'll see one by one step in our subsequent slides. Can you go to the next? As you know that those people are working in natural product or herbal drug standardization, they know that the extraction of herbal drugs is generally carried out by using conventional methods such as maceration, decoction, oxalate extraction, and so on. But nowadays, some green technological based methods are also using, such as supercritical fluid extraction, microwave assisted extraction, and so on. But in case of metabolomics, we require small amount of sample for characterization. Can you go to the next slide? As a result, in metabolomics, we generally extract sample we generally use liquid extraction technique. And if you see, there is no any universal method for metabolomic analysis. It changes, extraction, especially sample preparation method changes according to our interest of analyte. If suppose you are interested to analyze volatile compounds or polar compounds, for that you need to use different extraction method. As a result, sample preparation is considered as a fundamental and critical step in metabolomics, which define the outcome and accuracy of your result. So there are mainly solvent extraction is the common method used for sample preparation metabolomics, but according to your analyte or your matrix, we need to consider some additional steps like derivatization, sample cleanup by solid phase extraction is also routinely undertaken in sample preparation, metabolomic sample preparation. Generally, this derivation is used when we use GCMS, and in that case, derivation is important. And if you are using with biological, if you are working with biological matrices like plasma, urine, in that case, sample cleanup is very essential because matrix is very complex. So sample preparation is a really key step in metabolomics. Go to the next. The next step in metabolomics is data acquisition, which generally done for chemical profiling or fingerprinting as well. If you are using mass spectrometry for data acquisition, then we call it as a mass spectrometry based metabolomics. If suppose you are using NMR, generally proton NMR is preferred because it gives comprehensive information compared to carbon NMR. In that case, that sort of metabolomics called proton NMR based metabolomics. So both of these techniques are nowadays in use in metabolomics, but mass spectrometry is more preferred technique. As can you go to the next slide? The mainly because its sensitivity. If you see GCMS based or LCMS based metabolomics studies are more sensitive compared to NMR because these two techniques has more sensitivity. You can estimate or identify compounds those are present in pico and nanomole, but that is not possible with NMR. In NMR, only you can identify compounds if they are present in micromole quantity, micro to millimole quantity. So this is the main reason NMR mass spectrometry based metabolomics is more preferred compared to NMR based metabolomics. Though sample preparation is the another advantage you can say in case of NMR, but sensitivities nowadays is the most preferential criteria. That's the reason MS based metabolomics is more common. Can you go to the next slide? So based on aim of our study, or you can say aim of our analysis, generally two approaches are used in metabolomics, which is called targeted and untargeted. Untargeted is generally used to understand the data pattern in your study, or if you want to identify something novel compound. That's why it is called hypothesis generation. And this is generally non-quantitative studies, whereas targeted metabolomics, where targeted estimation of compounds, known compounds is carried out. But if you want to identify novel compound and if you want to prove the separation of one compound, one sample from other, or compare one compound with other, in that case, untargeted metabolomics is routinely used. In today's presentation, I will talk more about untargeted metabolomics because this is research-based approach, and it has a lot of significance in herbal drug standardization. So because targeted generally used to identify specific compounds, known compounds, so that can be an easy task. But if you see the untargeted, there are thousands of compounds and it is difficult to identify each and every compound. So can you go to the next? So here onward, that's why we'll see only untargeted metabolomics in detail. As I've said, there are 4 steps in metabolomics, whether it's targeted, untargeted. But first step is sample preparation. Next step is spectroscopic acquisition. That 2 steps are common. But if you want to do untargeted metabolomics, you need to have high resolution mass spectrometer or high resolution NMR, because we cannot use triple quadrupole because they have good sensitivity, but resolution is very poor. So when you are doing a mass spectrometry based untargeted metabolomics, we generally use top MS, orbit top MS, and for NMR based metabolomics, we use high gigahertz NMR such as 600 onward generally. Once you have data from mass spectrometer or NMR, that data we generally pre-process because we use that data for comparative analysis or future extraction or detection. So there are several online tools are available for pre-processing of data, such as MGMine, XCMS, MetaboAnalyst, and so on. That data we further analyze statistically by using various chemometric tools, especially to know the pattern in our data and if we want to identify chemical or biological markers. Can you go to the next slide? So data analysis is the key part of targeted metabolomics. For that, we need to use various chemometric tools. So chemometrics is not something that we are just using. These are the tools we are using for a long time. And chemometric means the science of extraction information from measurements made on a chemical system like GCL, CNMR, MS, NMR, with use of mathematical and statistical procedures. These techniques using in a lot of fields, including herbal drug standardization. Can you go to the next slide? Especially in untargeted metabolomics, we use two types of methods, you can say, as a chemometric tools to understand the chemical similarity and to identify feature. That is called supervised methods and unsupervised methods. Suppose you have two powders, they look similar, but just for looking, it's difficult to say that two sets of powder samples, they are similar or not. Whether they are chemically similar or not, we cannot say. So in that case, if you don't know the pattern, whether they are same or not, we use unsupervised methods. Generally commonly used unsupervised methods are principal component analysis and cluster analysis, just to compare the chemistry of two unknown samples. Once you know that these two samples are not same, then if you want to know why they are not same, then we use supervised methods. Means here we don't know, here we know pattern, but why that pattern is, if you want to know that time we need to use supervised method. Generally, two main methods are using in metabolomics is the PLS and PLSTA or OPLSTA. Can you go to the next? So now we'll see this methods, unsupervised method briefly. As I've said, most commonly used unsupervised method is PCA. PCA is the statistical method in which data is dimensionally reduced to identify a pattern in the data and to highlight similarities and dissimilarities. Can you go to the next slide? We'll not see these are mathematical terms, no need to go in detail, just to have an idea, because you will see this sort of words here onward. So generally there are two types of PCA plots are plotted. One is score plot and another one is loading plot. If suppose you want to identify the relation between samples, at that time we plot score plot, and if you are interested to interpret the relation among variables, then we plot loading plot. Can you go to the next? Next slide, please.
Just a minute, sir. It's not moving. Is it right, sir?
Yeah. So another unsupervised method in metabolomics commonly used is the cluster analysis. Generally, we use this method to identify pattern, to show relationship and group structure. Basically, this method. Hello. Hello.
Yes, Ravindra sir. Again, he will be sharing.
Sir, just a minute.
Meanwhile, I can try and I will try. I will share my screen and see how it goes. Can you see my screen?
Yes, sir.
Yes, sir.
Okay. So I was talking about cluster analysis. So as I said, it's a method generally used to identify patterns, relationship, and group structure. So among unsupervised methods, PLSDA, which is called partial least square analysis, is the most used or commonly used method. Its goal is to predict the set of dependent variables from set of independent variables. What it mean by dependent and independent variables are? Dependent variables are your treatments, or here you can see an experiment in which amount of water is added to understand its impact on phytoplant. So amount of water, whether if you are adding little or much more water, that is called independent variables. And its effect, it's called dependable variables. In general pharmacological experiment, our drug treatments is called as a independent variables, and effect of that drug, suppose hepatoprotective immune modulatory, that effect is called as a dependent variable. So this PLS method involves two steps: calibration step and prediction step. So I'll not go in detail. So the another supervised method, which is very important and more frequently used in metabolomics, is the PLSDA, or least square discriminant analysis, which is called PLSDA. According to some companies, they have their own algorithm as well, like OPLSDA, but there are similar kind. So aim of this use of this tool is to maximize covariance between independent variables and the corresponding dependent variables, means changes between the groups. And this technique has a lot of significance in untargeted metabolomics, especially to show the grouping as well as to identify the key feature responsible for that grouping. So we'll see one or two examples of each of this. So now we'll see example of one example of mass spectrometry and NMR based metabolomics, and I choose my own studies for these examples. And so first we'll see the example of mass spectrometry-based metabolomics. And for that, I would like to give example of my own study when I was in South Africa. Most of you may aware about propolis. So propolis is a resinous material produced by honeybees using bee wax and plant extrudants. And because it's a herbal based product, means a lot of plant extrudants are present in that. People have reported almost more than 800 different sorts of compounds in propolis. And several people have also reported that it has a very good biological activities such as antimicrobial activity, immunomodulatory activity. But one thing is sure that this is the only one effective natural antibiotic at the moment. So this propolis bees produce for their own defense, means for their own defense from other insects, microbes, and to maintain aseptic condition inside the beehives. Means this is, you can say, a natural selection of a bees of a drug. But problem with this propolis is its chemistry is highly complex. As I said, more than 800 compounds are reported in it. And its chemistry varies from region to region and season to season. Means because vegetation, if you see around the beehives, is always changing. And it changes with one region to another region. The plants which we see in Maharashtra, which we cannot see in Tamil Nadu, which we cannot see in Himachal Pradesh. So flora is also changing. That's why its chemistry is also changing. So characterizing this herbal product is really crucial and difficult as well. So what we did is to characterize propolis from South Africa, we use untargeted UPLCMS based metabolomics approach. And under this study, we collected around 39 South African propolis samples from 7 provinces. In South Africa, like India, they have we have state and they have provinces. So you can say we collected sample from 9 states of South Africa. And similarly, as I said, we use 4 steps of metabolomics to generate data. Initially, we collected sample, then sample processed using solvent extraction technique. Generally, here we used hydroalcoholic extracts to prepare samples, then analyze these samples by using UPLC Q-top MS. And after having that data, we process that data using SIMCA chemometrically. And here you can see PCA score plot, which clearly shows 2 groups among propolis samples collected from South Africa. And we also use 3 samples as the standards. Means we got some propolis samples from Brazil. If you see Brazil and South Korea, they are situated in South Hemisphere. So there could be possibility that the propolis samples in Brazil and from Brazil and South Africa could be similar. To understand that, we also analyzed that sample, and we found that Brazilian samples are totally distinct from South African samples. And then after that, we decided to understand why these 2 groups are present, or what are the key molecules. Because of that, these 2 groups are there. So that also we identified. We are generally identified almost 15 compounds in both the clusters, and we found, based on this study, we found that the propolis samples, including both types of propolis samples, are distinct from Brazilian propolis, means temperate zone propolis, and they have chemical similarity with the propolis samples we generally found in Europe, China, and so on. So temperate zone propolis samples. So after that, similar sort of work I did when I was in India. At this time, I use an NMR-based untargeted metabolomic approach to characterize Indian propolis sample. Under this study, what I did, I collected samples from almost 14 different states of India, from Jammu Kashmir to Tamil Nadu and from Gujarat to Bengal, and analyzed these samples by using proton NMR. And interestingly, I found that the propolis samples from India, they fall into two groups, and that grouping was region based, means those propolis sample found in northern part of India, they're totally different from propolis sample found in southern part of India. So we got clearly distinction between southern part propolis samples among southern part propolis samples and northern part propolis samples. And the key variations or key compounds responsible for this grouping were phenolic compound. So I used a targeted metabolomic approach, and I used various temperate and tropical zone propolis sample based chemical markers, and based on that identify the propolis in India is also have similarity with the propolis sample which found in China as well as Europe. So this studies, I believe, will help to commercialize this product. Suppose the European wants to propolis extracts, then they can take propolis from South Africa, they can take propolis from India. Even if suppose somebody wants propolis samples from somebody, means generally this propolis is used in cosmetics as well as a toothpaste, especially in Japan and South Korea, and they need very specific product, means they generally exporting from China. So we can sell that we can say that our product is similar to China, especially those propolis found in northern part of India, then we can sell based on this information. So that's the reason you can say this. You can see this chemometrics, mass spectrometry-based metabolomics has a lot of significance in this sort of generation of this sort of research data. So now we'll see some other examples of metabolomic studies which are used in herbal drug standardization, especially to understand the grouping among the samples or in biomarker discovery, and including quantification of specific chemical markers. First application of metabolomics we will see is related to quality control or developing fingerprinting profiling. These are the two Agostosma species, Agostosma betulina and Agostosma granulata. These both species are from South Africa, and they are traditionally known as a medicinal plants and used as a drug in various forms, as a antipyretic drug, antidiuretic drug, but mainly considered as a good drug for the treatment of urinary tract infection. However, if you see, both these species look similar, and if you want to identify which one is exact species, you need to have very good taxonomies, and generally that is not practically possible. So generally these species are taxonomically identified based on their leaf shape. So chemically differentiating these species, is it possible or not, that author studies in this study. So what they did, they used UPLCQ top MS untargeted metabolomics approach. Initially they created fingerprint LCMS based. This is the representative LCMS chromatogram at positive mode of Agostosma betulina and granulata. You can see this based on chromatogram, you can see there is a difference between these two species, but there are some compounds that are really common. So it's difficult to say whether this these two species are same or different by just looking chromatogram. So what they did further chemometric analysis of this data, and this is here at the right side of presentation. You can see PLS or PLSD score plot, where you can clearly see these two species are distinct from each other. Then they used S plot to identify which are the features are responsible for this grouping, and they identified quercetin as a marker compound from granulata and quercetin dimethyl ether glucoside for butylene. And they showed that this compound have ability to distinguish samples of Agostosma butylina and granulata. So after this, we'll see an example of how metabolomics can be valuable in identifying adulteration and contamination in a sample. So I'll give an example of a study where they used LCMS-MS based quantitative approach, or you can say quantitative metabolomics approach, for determination of non-opioid analgesic drug adulterants in herbal medicines. So what they did, this study is really interesting because in market we see there are a lot of herbal formulations for they're selling for painkiller, antipyretic as an antipyretic drug. But there is a chance that there could be contamination of modern medicines in that. So to understand same sort of thing, in this study, they collected samples from different market places, and they tried to identify the contamination of caffeine, acetophen, ibuprofen, diclofenac. And surprisingly, they found that there are several samples that had caffeine and other compounds. So this sort of adulteration definitely we can identify using LCMS or targeted based metabolomics approach as well. So geographical distribution, already I have given example of my studies on South African propolis, so I'll not explain it again. So now we'll see some of the examples, one or two examples of use of metabolomics in biomarker discovery. So similar to earlier studies, in these studies, they used two South African medicinal plants, Pelargonium species. These species have also a lot of significance in terms of medicine. It is routinely used for the treatment of upper respiratory tract infection in South Africa as well as in other African countries. But problem with this Pelargonium species, especially Pelargonium pseudois and Pelargonium reniforme, they look same, means even it's difficult for taxonomists to distinguish. And as a result, they are always unknowingly or knowingly adulterate and substitute to each other. Though both have medicinal value, but everyone wants to know which species they are taking as a drug. So in this study, they use untargeted metabolomics approach and try to identify marker compounds which can be used for their quality control. So they also created chemical profiles by using UPLC Q-top MS, and they showed that these two species have slightly different chromatogram. And you can see here in PCS score plot also, this species samples from these two species are totally different from each other, means they clearly form two groups, means they're different from each other. And then they identified the compound responsible for this grouping, and they recommend that compound for quality control of this species. That was Kelly was the key compound which found in pseudosis that can be used as a marker compound for quality control of Pelargonium pseudosis. So we'll see another example of biomarker discovery. This is a really interesting study as we are also eating tomato daily. So in this study, what they have did, they identified the biomarker of tomato consumption in mice compared to a lycopene feeding. And by using targeted metabolomics, they identified steroidal alkaloids in a plasma samples of mice, and they showed that that can be used as a biomarker of tomato consumption, because most of the time people are using tomato sauces, ketchups. So what they have ate, how we can say. So there should be some markers. So in the study, they identified steroidal alkaloids as a marker of tomato consumption. So with this, I would like to stop and thanks for your patience, though we had a lot of disturbance from my side as well as while sharing screen. So anyway, thanks for your presence and I'm happy to take questions.
Thank you, sir, for such a wonderful session. I hope I'm audible.
Yes, sir.
Yeah.
Yes. So thank you for such a wonderful session. So we have two questions here. So one question is that what are the opportunities for the export of medicinal plant raw materials from the Indian origin?
Yeah, definitely. If you see, Indian diaspora is everywhere in the world, and acceptance towards Indian things is also growing throughout the world. I will give an example of turmeric. If you see, many people here in US and in UK also, they really wanted to eat turmeric because they are thinking it's good for health. So, and I've seen some of the herb, as I've said, ashwagandha is very common in US. You will, if you go to any pharmacy in US, you will find ashwagandha. That mainly because it is considered Indian ginseng. That's why it's in market. Chinese and Koreans, they have promoted this their ginseng throughout the world, and they have made a lot of products. So I will suggest there is an opportunity for Indian products, but we need to sell our product based on scientific information. We need to provide more information about its quality. And then there is a lot of scope for Indian products, and we need to market our product as well, because Chinese, Koreans, they market their products like anything. I hope this answer will satisfy you.
Thank you so much, sir. There's one more question that we have here. How can we start for research related to metabolomics?
Yeah, basically, you need to know what sort of study you want to go. In our presentation, I said steps are simple, but you need to have what sort of metabolomics is not a single experiment. First thing we need to keep in mind, because in one experiment, we cannot tell everything, because metabolites are, their solubility is different, their other physical properties are different. Some of the metabolites are volatile, some of the non-volatile, some of the lipids. So we need to understand what sort of metabolomics we are doing. Suppose you are really interested to do metabolomics related to herbal drugs, that to only some small molecular weight compounds, then in that case you need to use specific instruments. For that, you don't need to have too much big instruments, but at least you should have some LCMS, MS or GCMS. Then based on that, you can initiate your metabolomics experiments, or you can plan your experiments. So first thing is you need to define what is your area of interest and what extent you want to go. Because sometimes for higher end of metabolomics, you need to have advanced mass spectrometers like TOF, Orbitrap, MS, and that is too costly to have. But with nominal GCMS also, you can start a metabolomics of small molecular weight compounds.
Thank you so much, sir. One more question we have is, in case we have no information on composition except preliminary screening, how can we use these tools, and how new compounds are identified that are not currently listed in the database?
Just as I said, if you want to identify a novel compound, you need to do a lot of studies. Just in one experiment, you cannot say that I have identified novel compound. Here in my lab, we are expert in metabolomics. We have our own library for compounds, and we have all sorts of mass spectrometers. Means we have 3 Q-tops, we have 3 Orbitraps, we have multi, everything we have. But still, we cannot say that yes, we can identify novel compound immediately in a one stroke. For that, we need to do a lot of experiments, and always means it's not a single experiment in that you can say that I have identified. You need to validate that compound as well. So first thing I will suggest, that after preliminary screening of your samples, you analyze that sample with GC or LCMS, convert that data, pre-process that data by using online tools. And once you pre-process that data, analyze that data statistically using various online tools such as meta-analysis. And if you find some interesting compounds, then check that compound, whether that compound present or reported in the literature or not. Or there are several databases also, online database, HMDB, HMQC, Phenol Explorer. So you can use that database and you can find whether your compound is novel or not. If you find still your compound is unknown and that is really significant, and then you can isolate that compound and further characterize and identify.
Okay, thank you so much, sir. One last question that we have is, while we are finding out the metabolomics process, does the process vary from the phytoconstituent category? Like if it is an alkaloid, if it is a glycoside, flavonoid. So does the process vary?
Yeah, that's what I said. No, that's why I said sample preparation is a key step in metabolomics. So according to your solubility of your compound, you need to design sample extraction, and you need to make variations. Suppose you are interested to analyze some essential oils that you cannot extract in a regular way. You need to extract in a proper way. So you need to understand what sort. First, you need to have an idea about what sort of analyte you are going to analyze. According to that, your experiment vary.
Okay, sir. Thank you so much, sir. Dr. Bhujbal, sir, are you having any?
Do I need to answer this meeting chat? I don't know. In chat section, there are some.
No. You can go ahead.
Okay, sir. Sir, I now request Dr. Maruti Shelar, sir, to conclude the session.
Thank you very much, Dr. Deepak sir, for inspiring all. At the outset, I would like to express deep gratitude for excellent session on application of metabolomics in herbal drug standardization. Sir, in your talk, issues with the herbal drug standardization, herbal medicinal global market, national scenario, and status of India in global market. Sir has also thrown the light on safety, efficacy, and most important part, that is quality of the herbal medicines. Sir also mentioned advances in the standardization of herbal drugs with special emphasis onto the metabolomics and different approaches of the metabolomics, like chemometric tools, PCA or PLS, as well as chemodiversity of South African propolis. Applications of metabolomics in herbal drug standardization, differentiation between the species, and how they are helpful for identifying the adulteration as well as the discovery of biomarkers. Last but not least, sir, you ended on a very positive note, emphasize the importance of advanced technologies in standardization of herbals. Your talk has given the lesson for the pharmacy teachers as well as researchers working onto the medicinal plants to give more focus onto the standardization of medicinal plant materials. At the end, sir, on behalf of Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune, we thank you, sir, for our humble invitation to being over here and sharing your knowledge and expertise and inspiring all. Thank you, sir. Thank you, one and all.
Thank you. Thank you so much for your attention as well.
Thank you so much, sir, for your for such a wonderful session. I now request the technical team to share the certificate. Sir, kindly accept this certificate as a token of appreciation. I feel honored and privileged to propose the vote of thanks for the session on behalf of the Institute. On behalf of the Institute, I would like to express my heartfelt gratitude to our guest speaker, Dr. Deepak Kasote, sir. I extend my heartfelt gratitude to Honorable Dr. P.D. Patil, sir, Chairman, D.Y. Patil Unitech Society, Honorable Dr. Somnath Patil, sir, Secretary, D.Y. Patil Unitech Society, respected Dr. Sohan Chitlange, sir, CEO Administration, Dr. D.Y. Patil Unitech Society and Principal DPU Pharmacy, Pimpri, Pune, and respected Dr. Santosh Putpal, sir, Vice Principal, DPU Pharmacy, for their constant support and inspiration and providing all the necessary facilities to conduct this event. I am also thankful to respected Dr. V. Edepalli, sir, Director Research and Professor, Department of Pharmacology, for his constant motivation and guidance. I would also like to thank respected Dr. Asha Thomas, ma'am, Professor and HOD, Department of Pharmaceutical Chemistry and Chief Coordinator of the event, for her constant support and guidance. I would extend my generous thanks to all the delegates for their cooperation and patient listening. I would also like to thank the entire organizing team and technical committee for their untiring support and in the smooth conduct of the session. The schedule for the upcoming session would be posted on the WhatsApp group shortly. Participants are requested not to leave the group until the completion of all the iFDP sessions. I now request all the participants to switch on their videos for a group photo.
Dr. Deepak, this is Dr. Asha. I would like to thank you very much, sir. It was an excellent session. And
Thank you.
You touched a very difficult subject area, which we usually do not look into, metabolomics and using all your tools to get to structure of the unknown compound. So it was great listening to you, sir.
No, actually, if you see, when we see as a India, means if you visit any pharmacy shop here in US, when I was in UK as well, so I saw that there is no any product from India, but other products from other country they're selling and people are purchasing. We need to do a lot of research for our products. We have a lot of potential, but we are not using our potential. I tried for Indian propolis, but definitely we need to work on others as well. And we need to have means whatever work we are doing, we need to be sincere in that. Then and then that work will benefit it to the society, as well as that will help to establish our brand in international market.
Absolutely, sir. Very right. Very right. Yes. So,
And my email is with organizers. You can, if you have any questions, you can directly ask me. Definitely, I can help you
yes, yes, sir.
if I
Yes.
can.
We will share the contact details, sir.
Okay.
Thank you, sir. Thank you very much.
Thank you.
So is it done or still we need to?
Yes, sir. Once again, I would like to just thank all of y'all for your kind attention. Thank you.
Thank you so much.
Yeah, thank you, sir.